So next we're going to be talking about the exceptions to Mendelian genetics. Inheritance of characters by a single gene may deviate from simple Mendelian patterns in the following situations. When alleles are not completely dominant or recessive. Second, when a gene has more than two alleles. And third, when a gene produces multiple phenotypes. Firstly, we will talk about incomplete dominance. So far, we have been observing complete dominance in the pea plant experiment. It occurs when the phenotypes of the heterozygote and dominant homozygote are identical. So when you have a dominant allele and a recessive allele, for example, the purple versus white plant, you're going to have a purple plant. The big P is going to be expressed instead of the little p. In incomplete dominance, the phenotype of F1 hybrids is somewhere between the phenotypes of the two parental varieties. In codominance, two dominant alleles affect the phenotype in separate distinguishable ways. So we will take a look at a couple of examples. So here we observe incomplete dominance in this species of snapdragon plants. The dominant trait is supposed to be the red flower. However, the red flower is not completely dominant. So when we have the two parents crossed together, the CW allele and the CR allele, and we observe the F1 generation, instead of the plants being red, we observe pink plants. This is because the CR allele is incompletely dominant. It is not going to be completely expressed over the CW allele. When we get to the F2 generation, by mating the F1 generation offspring, we get a different ratio than the 3-to-1 ratio we're used to. We get 1 red, 2 pink, and 1 white plant. So it's a 1-to-1 ratio. And this is because the heterozygotes are going to be showing the pink phenotype due to incomplete dominance. Next, we will look at an example of the relationship between dominance and phenotype. In the case of P-shape, the dominant allele codes for an enzyme that converts an unbranched form of starch in the seed to a branched form. So again, you remember capital R was round, that's the dominant allele, and lowercase r was wrinkled for the P's. Okay. So if you add the dominant allele right here, capital R, the phenotype is going to be the enzyme activity, and the enzyme is going to convert the unbranched form of starch into the branched form. Okay, so this is normal. However, in the recessive allele, which was little r, it codes for a defective form of the enzyme. So the enzyme is going to be defective, which leads to an accumulation of the unbranched starch, which is unable to be converted to the branched form. This causes water to enter the seed, which then wrinkles as it dries. Tay-Sachs disease is a fatal disease due to a dysfunctional enzyme that causes an accumulation of lipids in the brain. At different levels of organization, the mode of inheritance seems to be different. At the organismal level, the allele is recessive, At the biochemical level, the phenotype, which is the activity of the enzyme, is incompletely dominant, and at the molecular level, the alleles are codominant. Dominant alleles are not necessarily more common in populations than recessive alleles. You would think that dominant alleles, due to them being stronger than recessive alleles in their expression, would be more prevalent. However, for example, we have a condition called polydactyly. Polydactyly is a condition where a human would have extra fingers and toes. One baby out of 400 in the US is born with polydactyly, and it is caused by a dominant allele. However, polydactyly, the dominant allele, is much rarer and less frequent in the population than the recessive allele. So most people have the recessive allele. Most genes exist in populations in more than 2 allelic forms. So opposed to our 2 allele examples in the pea plants, we have an example of blood type. Blood type is going to be including multiple alleles. There are 4 phenotypes of ABO blood groups in humans, and they are determined by 3 alleles for the enzyme that attaches A or B carbohydrates to red blood cells. The blood type A allele is IA, B is IB, and the type O is lowercase I. The enzyme encoded by the IA allele adds the A carbohydrate, whereas the enzyme encoded by the IB allele adds the B carbohydrate. The enzyme encoded by the lowercase I allele adds neither. So here we see the different carbohydrates that are added to the surface of the red blood cell depending on the allele the individual has. Here are the different possible genotypes. If you have a AA or a IA lowercase I, then you will be blood type A, having A carbohydrates on the surface of your cell. If you have IB, or IBO, you will have B carbohydrates on the surface. But notice right here, if you have IA and IB, the A and B are going to be equally expressed and equally present on the surface of the cell. This is also an example of something called codominance. The A allele and the B allele are equally dominant, therefore they're both going to be equally expressed as a phenotype. If you have lowercase I, or OO, the red blood cell will have neither carbohydrates on the surface of the cell. The next exception is pleiotropy. Most genes have multiple phenotypic effects, a property called pleiotropy. For example, pleiotropic alleles are responsible for the multiple symptoms of certain hereditary diseases, such as cystic fibrosis, which is a disease causing sticky mucus, and sickle cell disease, which is a disease that causes misshapen red blood cells that look like sickles or crescents. Some traits may be determined by 2 or more genes. In epistasis, one gene affects the phenotype of another due to interaction of their gene products. In polygenic inheritance, the word poly meaning several and genic meaning genes, multiple genes independently affect a single trait. In epistasis, expression of a gene at one locus alters the phenotypic expression of a gene at a second locus. For example, in Labrador retrievers and many other mammals, coat color depends on 2 genes. One gene determines the pigment color, with alleles big B for black and little b for brown. The other gene, with alleles big E for color and little e for no color, determines whether the pigment will be deposited in the hair. If heterozygous black labs, genotype big B, big E, little e, are mated, we might expect the dihybrid F2 ratio of 9:3:1, as we saw earlier. However, a Punnett square shows that the phenotypic ratio will be 9 black to 3 chocolate to 4 yellow labs. Epistatic interactions produce a variety of ratios, all of which are modified versions of 9:3:1. So here we see the departure from the 9:3:1 ratio we would expect in a dihybrid cross due to epistasis. The second gene, gene E, is going to affect whether gene B is going to be expressed or not. As you can see here, the black dogs all have a dominant allele for gene E. And they are black because they have a dominant allele for gene B. On the other hand, if you look at the brown dogs, they also have a dominant allele for gene E. And so gene B can be expressed. And since they have a recessive allele for gene B, they are brown. Let's move on to our yellow or pigmentless labs. As you can see, they do not have a dominant allele for gene E. They have recessive alleles. This is going to prevent the depositing of the pigments. So it does not matter whether you have a dominant allele for gene B or a recessive allele for gene B. They are all going to be the same color because gene E is going to influence the phenotype or the expression of gene B because of epistasis. Quantitative characters are those that vary in the population along a continuum. Quantitative variation usually indicates polygenic inheritance, an additive effect of 2 or more genes on a single phenotype. And so because of this, we have a range of diversity or variation. Height is a good example of polygenic inheritance. Over 180 genes affect height. So it's not one gene, but 180 genes, making it a lot more complex. Skin color in humans is also controlled by many separately inherited genes. So here we can see how multiple genes, gene A, gene B, and gene C, are going to affect the skin color, or which shade of skin color you have. And due to having multiple genes determining a single phenotype, we can have a lot more combinations or a lot more variety of traits. Another departure from Mendelian genetics arises when the phenotype for a character depends on environment as well as genotype. The phenotypic range is broadest for polygenic characters. Traits that depend on multiple genes, polygenic, and also combined with the environmental influences are called multifactorial traits. This is an example of a multifactorial trait. The hydrangea plant, depending on the pH of the soil that it's grown in, will have a different flower color. Hydrangeas that are grown in basic soil will have a red color, while hydrangeas that are grown in acidic soil will have a blue color. So the environment is affecting the phenotype. An organism's phenotype includes its physical appearance, its internal anatomy, physiology, and behavior. The phenotype does not always have to be expressed on the exterior, but can be expressed in the interior. An organism's phenotype reflects its overall genotype and unique environmental history. Humans are not good subjects for genetic research because generation time is too long, and also parents produce very few offspring compared to other organisms, and breeding experiments are unethical. However, basic Mendelian genetics endures as the foundation of human genetics. Finally, we will discuss pedigrees. A pedigree is a family tree that describes the interrelationships of parents and children across generations. Inheritance patterns of particular traits can be traced and described using pedigrees. Pedigrees can also be used to make predictions about future offspring. We can use the multiplication and addition rules to predict the probability of specific phenotypes. So here we see two examples of pedigrees following two different traits. First, we want to take a look at what the shapes and the lines represent. The square is going to represent males. The circle represents females. If the shapes are shaded in, it means that the individuals have the trait. And if there's a line that's connecting the two shapes together, it means that they both have mated. The offspring are going to stem from the parents, and the firstborn is going to be on the furthest to the left, and the lastborn is on the right. So let's take a look at our first example. We're going to follow a trait called the widow's peak, which is a dip from the hairline on the forehead, and it is a dominant trait. So if we have capital W, it means that you have the widow's peak, or that's the widow's peak allele. If you have the recessive allele, then you do not have widow's peak. So we remember that we can have three different genotypes. If you have a heterozygous genotype, you still will have the widow's peak due to the law of dominance. And that's what we see here. For the first generation, who are the grandparents, the male is going to be heterozygous, but is still going to express the widow's peak due to the law of dominance. The female is homozygous recessive, which means that they don't have the widow's peak. They both mate together and have 4 children. 2 of the children do not have the widow's peak, and 2 do have the widow's peak, which is 1 male right here. And we can see how the male from this family mated with the female from another family. And the female also had the widow's peak. They are both heterozygous, and they mate together to have 2 children. And one of the children has a widow's peak, and the other child does not have a widow's peak. And so we can see these 2 children and the phenotypes that are observed. Okay. The example to the right shows you a trait called PTC tasting. So the trait is the ability to taste PTC. And this is also going to be a dominant trait. So if you have capital T, you're able to taste PTC. If you have the recessive allele, you cannot taste PTC. Okay. So if we follow along from the first generation, we can see 2 families here. The grandparents, the first generation. So we have 2 individuals who can taste PTC. However, we can look right here. Because they're heterozygous, they still carry that recessive allele. So 2 of the individuals are not able to taste PTC, but the other 2 individuals are. Because they are they have at least one dominant allele. So this male is going to mate with another female from another family, and they both are able to taste PTC, but they have two children, one who is not able to taste PTC and the other who is able to taste PTC. So in this case, it seems like being unable to taste PTC is the trait that we're following. And in the other pedigree, the widow's peak is the trait that we are following. Many genetic disorders are inherited in a recessive manner. These range from relatively mild to life-threatening. Recessively inherited disorders show up only in individuals homozygous for the allele. So you have to have both recessive alleles. Carriers are heterozygous individuals who carry the recessive allele but are phenotypically normal. Most individuals with recessive disorders are born to carrier parents. Albinism is a recessive condition characterized by a lack of pigmentation in skin and hair. So here we're going to follow a cross between two parents. And on the right, we can see a child with albinism, a condition where the skin cells are unable to create melanin, which gives color to your skin. The parents both are normal. They do not have the albinism phenotype. However, they are carriers. And that's because albinism is a recessive trait. Okay. So if we have them mate together, we're going to have a 1 out of 4 chance that we will have a child with albinism. And we have a 2 out of 4 chance of getting a normal phenotype individual, but they are carriers. And we have a 1 in 4 chance that we will get a normal phenotype individual that is not a carrier. If a recessive allele that causes a disease is rare, it is unlikely that two carriers will meet and mate. Consanguineous matings between close relatives increase the chance that both parents of a child carry the same rare allele. Most societies and cultures have laws or taboos against marriages between close relatives. Cystic fibrosis is the most common lethal genetic disease in the United States, striking 1 out of every 2,500 people of European descent. The cystic fibrosis allele results in a defective or absent chloride transport channels in plasma membranes, leading to a buildup of chloride ions outside the cell. Symptoms include mucus buildup in some internal organs and abnormal absorption of nutrients in the small intestine. Sickle cell disease affects 1 out of 400 African Americans. It is caused by the substitution of a single amino acid in the hemoglobin protein in red blood cells. So substitution is a mutation where we replace the normal amino acid with another amino acid. And this is caused by a substitution that happens at the DNA originally. In homozygous individuals, all hemoglobin is abnormal, sickle cell. And I think it's good to be specific homozygous recessive individuals because sickle cell disease is a recessive disease. Symptoms include physical weakness, pain, organ damage, and even paralysis. And Heterozygotes said to have sickle cell trait are usually healthy but may suffer some symptoms. About 1 out of 10 African Americans has sickle cell trait in unusually high frequency. However, there is one beneficial aspect to being a heterozygote. Heterozygotes are less susceptible to the malaria parasite. So there is an advantage to being heterozygous in regions where malaria is common. So here we see a diagram of how that substitution mutation is going to lead to the sickle cell disease trait. So here we have a heterozygote and a homozygote. This individual is going to have a substitution mutation in the DNA in the chromosomes of both the paternal and the maternal chromosomes. This is going to lead to sickle cell hemoglobin proteins. Hemoglobin is a protein in your red blood cells that is important for carrying oxygen. So when the hemoglobin protein is compromised, it leads to an inability or low ability to carry oxygen. The mutation and the change in the hemoglobin protein shape is going to cause a change in the shape of the red blood cell from a round red blood cell to a sickle-shaped red blood cell. This is going to cause systemic events. It's going to cause several symptoms throughout your body, which is, as we saw earlier, an example of pleiotropy. In the picture below, we have a heterozygote. They have one sickle cell allele and a normal allele. Some of the hemoglobin in some of the red blood cells are normal, and others are not normal. This leads to a mixture of sickle cell red blood cells and also normal red blood cells. And also notice right here at the top and the bottom, for individuals that are heterozygote, they need to have, or they need to be in environments with very low oxygen to show symptoms. Some human disorders are caused by dominant alleles. Dominant alleles that cause a lethal disease are rare and arise by mutation. Achondroplasia is a form of dwarfism caused by a rare dominant allele. Here are two parents. One has the dwarfism trait, achondroplasia, and the other parent is normal, being homozygous recessive. It seems like the father is the heterozygous individual. So we have the father, the sperm at the top, and the mother's gametes on the left. We cross them together, a heterozygote with a homozygous recessive individual, and we get two dwarf, or our chances of getting dwarf offspring is 50%, and normal offspring is 50%. And on the right, we can see the phenotype for dwarfism. The timing of onset of a disease significantly affects its inheritance. Huntington's disease is a degenerative disease of the nervous system. The disease has no obvious phenotypic effects until the individual is about 35 to 40 years of age. Once the deterioration of the nervous system begins, the condition is irreversible and fatal. Many diseases such as heart disease, cancer, alcoholism, and mental illnesses have both genetic and environmental components. No matter what our genotype, our lifestyle has a tremendous effect on phenotype. So this brings us back to the multifactorial traits or multifactorial disorders. Genetic counselors can provide information to prospective parents concerned about a family history for a specific disease. So how can genetic counselors help? Hypothetically, let's say that a couple both have a brother who died from the same recessively inherited disease. A genetic counselor can help determine the risk that the child of a couple will have that particular disease. It's important to remember that each child represents an independent event in the sense that its genotype is unaffected by the genotypes of older siblings. If both members of the couple had a sibling with a recessively inherited illness, both of their parents were carriers, assuming that the parents were normal. Thus, each has a 2/3 chance of being a carrier. If both are carriers, there is a 1/4 chance of each child having the recessive illness. The overall probability of them having a child with the illness is 2/3 times 2/3 times 1/4, which equals 1/9. Okay. So if both of their parents were carriers, let's assume that we're taking a look at a recessive disease because there are carriers for the disease. So the dominant allele is normal, and the recessive allele is the disease. So if both parents originally were carriers, that means that they're heterozygous. Okay. Okay, so if we do a Punnett square right here, we're going to put the heterozygotes on the left and the right. And we're going to get this ratio here, our 3:1 or 1:1. So you can see how because the husband and the wife both had siblings that had the disease, and because they don't have the disease, well, we're only gonna take into account the homozygous dominant and the heterozygous. So two of them are going to be heterozygous, so the chance is 2 out of 3. Okay. So thus each has a 2/3 chance of being a carrier. Okay. Again, if both are carriers, there's a 1/4 chance of each child having the recessive illness. So it's the same thing right here, 1/4 chance. So if we made it both the carriers for the second generation, so assuming that they're both carriers for the couple, it's going to be the same Punnett square. There's only a 1 in 4 chance of an affected child. Okay. So the overall probability of them having a child is, what's the probability of both of their parents being carriers? Well, for one of the one person as okay, if one of the people who made it is 2/3 and the other person is 2/3, the husband and the wife, and also we multiply it by their chances of getting a affected child, we just multiply them. So if we put this into words, it's basically saying what's the probability of one person being a carrier and the other significant other being a carrier, 2/3, and the child being affected with the disease. Okay, so you just multiply them and get 1/9. So another way you can look at it is for each multiplication sign, those represent the word and. Okay, what's the probability of this and this and? What's the probability of being a carrier for the female and the carrier for the male and getting a affected child? Okay, if we worked out the math, 2/3 times 2/3 times 1/4 equals 4. 2 times 2 times 1 is 4. Over 3 times 3 times 4 is going to be 9 times 4, which is equal to 4 over 36. And we reduce that to 1 over 9. Okay. For a growing number of diseases, tests are available that identify carriers and help define the odds more accurately. The tests enable people to make more informed decisions about having children. However, they raise other issues, such as whether affected individuals fully understand their genetic test results and how the test results are used. In amniocentesis, the liquid that bathes the fetus is removed and tested. In chorionic villus sampling, CVS, a sample of the placenta is removed and tested. Other techniques, such as ultrasound, allow fetal health to be assessed visually in utero. So we have amniocentesis and chorionic villus sampling on the right. So you can see how they're extracting the samples and basically analyzing the cells, allowing us to get a karyotype of all the chromosomes, which includes the genes. This allows genetic counselors to assess the risk of a child inheriting a disease. Here's an ultrasound of the human fetus. Some genetic disorders can be detected at birth by simple tests that are now routinely performed in most hospitals in the US. One common test is for phenylketonuria, PKU, a recessively inherited disorder, which is the inability to metabolize an amino acid called phenylalanine, which is found in many proteins and artificial sweeteners. This disorder occurs in 1 of every 10,000 to 15,000 births in the US, so it is very rare.